Hey guys, welcome back to the channel. So in this video we are going to talk about plasmids and restriction endonuclease, which is a part of genetic engineering. So let's get started. So in this video we'll discuss about plasmid DNA, which is also known as a cloning vector. So in my last video I discussed about recombinant DNA technology, in which this particular plasmid DNA has a major role for formation of recombinant DNA. So I'll discuss about what is plasmid DNA and what are the features of plasmid DNA so as to why we use them in the recombinant DNA technology. So plasmids are nothing but these are circular molecules of DNA that lead an independent existence in the bacterial cell. Also plasmids almost always carry one or more genes, and often these genes are responsible for a useful characteristic displayed by the host bacterium. Also, plasmid DNA can be used as a cloning vector, as said before. So, some of the features that makes it useful, and it makes it useful for using it as in a recombinant DNA. So, here we go. So, some of the features are selectable marker. So, some of the examples for selectable markers are antibiotic resistant gene such as chloroamphiphenical, amphipenicillin, streptomycin, erythromycin, tetracycline, etc. resistant gene. So next we have multiple cloning site, which is the cloning site for gene of interest. So this particular has a recognition site of many different restriction endonuclease enzymes. So in my next video I'll be talking about restriction endonuclease enzymes so as to you can correlate the plasmid DNA with restriction endonuclease. Also, it has another feature which is origin of replication. So most plasmids possess at least one DNA sequence that can act as an origin of replication, so they are able to multiply within the cell independently. So these are some of the important features such as selectable marker, which is helpful for detecting the recombinant DNA or the ones which have transformed with the help of some of these resistant genes. We have multiple cloning sites for the gene of interest for the particular DNA strand or the gene we are looking for, and then we have the origin of replication, so which matters the most, where the DNA multiplies. So with that, so I'll show you a pictorial view of how it looks like. So this is a basic plasmid or a cloning vector, you can say. So this is how it looks. So this is the antibiotic resistant gene, or this is the place where we can find these particular genes for identification of recombinant ones, recombinant DNA one, recombinant or transformed cells, and then we have the multiple cloning sites, and then we have the replication site or the origin of replication. So let's just keep this video till here. Hope you enjoy this video and stay tuned, and I'll be back with another one.